Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their projects and data, highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen, Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, it is my pleasure and privilege to welcome Connie Cepko as our speaker. Her talk is entitled Gene-Agnostic Therapy to Preserve Vision. Connie is Bullard Professor of Genetics and Neuroscience at Harvard Medical School, Professor of Ophthalmology at the Massachusetts Eye and Ear, and an investigator of the Howard Hughes Medical Institute. She received her Ph.D. from the Massachusetts Institute of Technology, working with Nobel laureate Philip Sharp, and completed postdoctoral training with Richard Mulligan, where she helped develop retrovirus-mediated gene transduction techniques widely used to study gene function in the nervous system. Her laboratory has made seminal contributions to understanding cell fate specification and circuit formation in the vertebrate retina. She now focuses on mechanisms of photoreceptor degeneration and the development of gene-agnostic gene therapies to prolong vision. Dr. Cepko is member of the National Academy of Sciences and the American Academy of Arts and Sciences, and has received numerous awards recognizing her research and mentoring in developmental neurobiology and vision science. Before you start, Connie, let me remind the audience that you will be able to ask questions throughout the event and also give a thumbs up to other questions. We will get to those questions at the end of Connie's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. The Q&A panel on the right-hand side will stay available for a couple of days after the live event. So if you are currently not watching live, but a recording of the event, and if Q&A is still available, please enter your name and your question so that we can back to you with the answer. We are very much looking forward to your questions and a lively discussion at the end. So Connie, I ask you to unmute yourself and share your screen with us. And I'm very much looking forward to your talk.
Okay, can you see my screen?
Yes, we can.
Okay. Well, first, thank you very much for the invitation.
No, not yet. You still have to share your screen.
Okay.
Still not.
I hit the share button and then I hit share the window. Okay, try that again.
There we go.
You can see it now?
Yes, thank you so much.
Sure. Sure. Well, first of all, thank you very much for the invitation to speak with you today, and also for the use of your optodrum, which has been really great for several of our studies, as you're going to see in the talk. But I'd like to start with this first image. This is an image of a cone photoreceptor in the mouse. It was reconstructed by Claudio Punzo when he was a postdoc in the lab. And what I like about it is it shows some of the important features of a photoreceptor. So this green area is the outer segment of the photoreceptor where phototransduction takes place. Light is captured and the signal is processed. The red little spot here is part of what's called the inner segment, and the red stain is from phosphorylated mTOR. So phospho-mTOR indicates that this is a healthy cell. It can carry out metabolism. And then this magenta area is where the nucleus and cell body is of the photoreceptor. There's actually very little cytoplasm here. Most of the business of the photoreceptor takes place in the inner segment and the outer segment. So this is the structure that we're trying to preserve with our therapies. One of the first things that happens when a photoreceptor starts to lose function is this outer segment degenerates. At that point, the photoreceptor is usually not very active in terms of capturing and processing light. So we're trying to regrow it and keep the cell alive as long as possible using our therapies. So just to make sure we're all on the same page, this is just a diagram of a vertebrate eye. And the retina, the neurosensory part, is this thin yellow line at the back of this globe. It's only about 200 microns thick. And in humans and birds of prey, some lizards, we have a high acuity area located in the macula, which is virtually in the center of the eye. The highest acuity part is actually in the center of the macula. That's the fovea. It's only about 1% of the surface area, yet it provides about 50% of the input from the eye to the brain. So it's a very important area for us. And for some reason, it's vulnerable to degeneration, as in age-related macular degeneration. And I'll be talking about that at the end. But the first part of the talk will be about diseases that affect the entire retina, and some of the diseases that you may have heard of, like retinitis pigmentosa. So this is just some of the genes that lead to loss of vision. These have been named historically by the ophthalmologists who studied them. On the top left, you see retinitis pigmentosa genes. It's just a subset. We're actually over 100 genes that can lead to this disease. And I'll describe the disease to you in a moment. But you can see there's other diseases listed here, like cone rod dystrophy, Usher syndrome. And these also lead to loss of vision, and in some cases have additional symptoms like loss of hearing. And we're hoping that if we have a therapy for retinitis pigmentosa, it will also be useful for some of these other diseases. And you'll hear at the end, perhaps age-related macular degeneration. But our thought was that there's too many individual genes to address each one with something like gene editing or gene augmentation. Most of these genes, when they're mutant, only are present in a very small number, actually, relatively small number of people, making it really impractical for a company to address each of them in a clinical trial, which is very expensive. So when we saw the number of genes, and actually there's now over 200 for the whole set of genes that lead to loss of vision, our thought was we need to come up with a strategy that will help multiple families from a single intervention. So retinitis pigmentosa is a disease where a person is born with full field, high acuity color vision, as in this image, and the person then starts to lose their color vision. Oh, sorry, I should say, and they don't have good night vision. They start to then lose their color vision as well as their night vision, first from the periphery, and it can proceed all the way to the central high acuity vision, so a person can lose all vision. Now, we can think about this disease with this following cartoon. So again, a person is born with poor or no night vision, and that's because the rod photoreceptors, which are the most abundant photoreceptors, are not functioning due to some type of disease gene expression in the rods. And most of those disease genes for retinitis pigmentosa are only expressed in the rods, which explains why the first symptom is loss of night vision. But as a person ages, then they can actually lose the rods, they will die, and then the cones start to die as well. And that means that a person will lose their daylight vision, their high acuity and color vision, which is, we need the cones for that. So first loss of night vision, followed by loss of cone vision. Now this cone vision loss has to be due to what we call non-autonomous or bystander effects, because the cones don't express the disease gene. So what's going on that leads to loss of cones? That's one of our major questions, because if we could address those causes, and if they were in common across different disease gene families, we may be able to address them with a gene-agnostic strategy. So that's what we've been doing. We're trying to understand, why are these cones dying? Now, there's a number of possibilities in the literature and from our own work. One possibility is that the rods make a trophic factor necessary for cone survival. Upon loss of the rods, then we lose that trophic factor. And there is some evidence for that from Jose Sahel. He has a factor called rod-derived cone viability factor, which does provide some support for this theory. Other theories have been that there's toxins released by the rods when they're dying. I don't think that's true given the kinetics of the little cartoon here. We actually measured the kinetics in 4 mouse models of retinitis pigmentosa. And what's represented here is what we found. That is, the cones don't start to die until almost every rod in their immediate neighborhood dies. Now, if there was a toxin elaborated, we think that would happen sooner. So at the peak of rod death, that would be the peak of toxin concentration, then the cone death would start sooner. And that's not what we see. It's quite delayed, which is great. It gives a large window of time for treatment. In our own work, we found there's metabolic problems. I'll talk to you about that. And then in practically every disease, there's inflammation and oxidative stress. And again, I'll talk to you about those. So with this list, then, we thought maybe we could address these different problems by introducing an AAV gene therapy vector carrying a disease-fighting gene. And I'll tell you about those. So again, the goal here is to slow down or prevent cone death and administer this therapy once a person starts to lose their daylight color vision. So the metabolism I mentioned is something that we came up with when we did RNA-seq across these 4 mouse models of retinal degeneration. We asked what's in common in terms of gene expression changes when the cones start to get sick, and it pointed us to metabolism. So here's just a little cartoon explaining the interchange between the photoreceptors and their support cells here in gray, the retinal pigment epithelium. So the photoreceptors are apparently one of the hungriest cell types in the body. They use a lot of glucose. They also have high concentration of mitochondria. They also are in a high oxygen environment. And they get their glucose, which is their main fuel, from the choriocapillaris, which is not shown here, but it's a blood vessel layer just on the outside of these RPE cells. So glucose comes from the choriocapillaris into these RPE cells and then flows into the photoreceptors. They then carry out glycolysis, and as a product of glycolysis, they secrete lactate, which goes back into those RPE cells. The RPE cells then use the lactate for their own fuel, which allows them to give up the glucose to the photoreceptors. So this little interchange, glucose goes in, lactate comes out of the photoreceptors, is what's working in a healthy retina. Now we have to consider what happens when the rods die. Now the rods, again, are 95% of the photoreceptors, which means when they die, there's very little lactate production now coming out of the retina, which means the RPE is not getting its primary fuel. So it may hold on to the glucose and not give it up to the remaining photoreceptors, which are mostly cones. So we think then that the cones are degenerating because they're not getting their preferred fuel, which is glucose. So this is a model that the field has supported with a variety of tests. It's not actually established by what I hope someday will be real live imaging of these metabolites in an in vivo setting, but we have some workarounds, and we have some ways to address this model. So our first thought was, well, if cones aren't getting enough glucose due to the retention of the glucose in the RPE, can we get a better supply for the cones? So we have a couple strategies for that. One is to have them or the RPE make glucose, which they do not ordinarily do. Another was to give the RPE more lactate to make up for the lack of lactate coming out of those dying rods. And the other strategy was to induce the cones to use more lactate. Regardless of where it comes from, perhaps Mueller glia give it to them. There may be other ways for them to get lactate, but have them use the lactate as their primary fuel rather than wait around for more glucose. Now, I'm not going to have time to tell you all of these things, but I'll tell you a little bit of these. First one was to have them or the RPE make more glucose. So the way we do our experiments is we create these AAV viral vectors and we put into them genes that we hope will allow them to make the photoreceptors survive longer. We also put in a gene, usually GFP, so we can track where the inoculum goes. And we're using doses that are high enough that we usually get co-infection with all of our vectors. We inject at postnatal day 0 in the subretinal space, which allows the virus to get into the photoreceptors and the RPE. And we do this early on because at that time, we get two benefits. One is the inoculum flows between the photoreceptors and the RPE without the impediment of outer segments and RPE apical processes, which will develop later. And those processes will then not permit the flow of the inoculum throughout. But at this age, there's no block. We get full infection of the RPE and the photoreceptors. Also, at this age, we are damaging the eye when we inject, and it tends to heal quite nicely. So we deliver these viruses, and we usually use 3 different models of retinitis pigmentosa in mice, because again, we're trying to get a gene-agnostic therapy. So whatever we develop, we want it to work across disease gene families. So we let the animals grow, and these are models of retinitis pigmentosa with different kinetics of vision loss, photoreceptor loss, and we then take the animals at different stages in their degeneration. We take out the retina, flatten it out, we count the remaining cones. We also can take out the RPE and again count the RPE cells. So we're quantifying now typically in the center because that's where the first degeneration happens. And importantly, we also want to assay visual function, and for this we use the optodrum. So we're getting measurements of acuity in the live animals, and we can do that over time. So our first strategy was to have the RPE and the cones get the capability to make their own glucose, which again is not really something most cells do. Mostly this happens in the liver and a little bit in the kidney. And what's shown here in blue is the glycolytic pathway. So I think most of us know from basic biochem that glucose comes in, it goes through a number of enzymatic changes to create pyruvate that can be converted to lactate. But in the liver, it goes the opposite direction. It uses some of those blue enzymes, but there's 3 additional enzymes, FBP1, PepCK, and PCX, which allows the reaction to go backwards, starting out with lactate or other amino acids that are called glucogenic amino acids, and they can then reverse to make glucose. That's gluconeogenesis. So we thought, well, if there's not enough glucose, but maybe there's enough lactate or other amino acids that could serve as fuel for this pathway, we'll put in these 3 enzymes, again, using AAV, and see if the cones live longer. And they did. So these are counts now of the cones in the center and the periphery. We're counting GFP cells because, again, we're using GFP as a marker for infection and also a control. So if we put in just GFP, we have very few cones left at this time point, either in the center or the periphery. Now look at the pink dots. These are when we're giving gluconeogenic enzymes along with the GFP, and you can see we have greater survival of cones in the center or the periphery. We also have a treatment I'm going to tell you about called NRF2. It's a transcription factor that fights oxidative damage. And it also has a nice rescue of cones. And the important point is when we put the 2 together, the gluconeogenic enzymes and the NRF2, we get an additive effect. We get a boost. And the reason we think we're getting the boost is, as I mentioned in the beginning, we think there's oxidative damage and lack of glucose. So having the 2 together is remedying 2 problems that the cones have. And it's nice to see it's additive. And I'll just draw your attention to this bottom set of panels. This is the optomotor assay. So in pink is gluconeogenesis, and green is just GFP alone. And on the x-axis is the y-axis is visual acuity, and x-axis is age. And you can see the animal is losing acuity over time in this model. So this is one model, and here's another model on the right. And so you can see that we're getting retention of some of the acuity, even though we are losing it over time. And we think that we're not getting a really stable rescue because there's still other problems like inflammation that we haven't dealt with this combination of AAVs. So we believe we're going to have to do more than one thing to really get a stable rescue. Okay, so that was asking the cells to make their own glucose. The second thing was to ask the RPE cell to take up more lactate. And we're wondering, why isn't it taking up more lactate? Because there is lactate in the blood, and they have lactate transporters. If they're not getting it from the rods, that's their apical surface, why are they not taking up more from the blood? It might be because their lactate transporters are not high enough affinity. So this is a little cartoon. The RPE is in gray. The blood is now shown in red here. And again, glucose can come out of the blood, go through glucose transporters and flow into the photoreceptors. Lactate can do the same thing, and lactate comes into the RPE from the photoreceptors with this yellow one here. If we look at the lactate transporters, the purple one and the yellow one, we see that they don't have a very high affinity. So we get KDs of about 3 to 5 or 6. Blood lactate's only about 2.5 to 4.6 millimolar. So it isn't going to allow a lot of uptake through these transporters. But there is a lactate transporter that is much higher affinity, and that's the MCT2 transporter, which is not normally expressed in any of these cells. So we thought, well, if we gave the RPE MCT2, maybe that will encourage it to take up more lactate from the choroid and thereby let the glucose go back to the cones. So Laurel Chandler, a postdoc in the lab, did exactly that. She gave these different models, AAVs with MCT2, and we found we did get greater cone survival and greater acuity. So these are just some flat mounts showing you the cones. These are the counts. And you can see a nice boost 2 to 3-fold with the MCT2 in one model, and this is in another model. And with optomotor measuring with the optodrum, we also got an increase. Again, in red is at 40 days of age, and here's at P53. Again, it wears off, sadly, but we still do have a boost at both ages. So better cone survival, better acuity. Now we wondered what's really happening in those cones when we give, or sorry, in the RPE, when we give them this MCT2 transporter. So there's a family of chemogenetic protein-based FLIM biosensors. Some of you may have heard of them. They're these really clever designs. People have found proteins, usually in bacteria or fungi, that bind small molecules like glucose or lactate. And you can fuse them to a fluorescent proteins like GFP. And what's diagrammed here is when one of these fusion proteins finds its little metabolite that it binds to, it can change conformation and thereby change its fluorescent output. So these are called fluorescent lifetime images. And what's being measured, you can get a microscope to measure how long is this fluorescence for, because the fluorescence will then decay. And the decay rate is going to depend on the concentration of the little metabolite. And that's kind of diagrammed down here. So this is time in nanoseconds for the photons being emitted by this fusion protein when it's bound to its metabolite. So more metabolite is usually more lifetime. It's not true for all of them. I'll say the lactate one does the opposite. When it's not bound, it has a longer lifetime. But the glucose one that I'll show you has a longer lifetime when its metabolite is bound. And in fact, that's how most of them work. So we made an AAV that encoded 2 of these sensors, one for glucose, one for lactate. And what I'm showing you here on the top set of panels is a control infection with GFP and a sensor. And on the bottom set of panels is infection with the MCT2 and a GFP and a sensor. I'm sorry, not GFP in this case, just the sensors. The sensors have the fluorescence. So we can measure then the lifetime when we add lactate to the media. These are immediately explanted after being infected in vivo. We wait a couple days, we take out the RPE, we flatten it out, and we immediately do imaging. And we look at what is true in terms of lactate concentration before we add any or after we start adding some to the media. And what you can see here is that we change the lifetime. So in the beginning, which is the most important part of this panel, we have more lactate in the MCT2 infected retina, or sorry, RPE than the control. So this tells us that the MCT2 led to higher intracellular concentration of lactate, just what we hoped. Now, it can also continue to add more and more if we add it to the media. Again, if the MCT2 is able to take up lactate, we should get a higher value, and we do. These are graphed down here. So the MCT2 is always giving us more of the signal indicating lactate is present than the control. Now, the control can also take up lactate, which makes sense because I told you that the lactate transporter with lower affinity is present in the normal RPE. So this was really great for us because we had cones living longer, we had greater acuity, and indeed the lactate seems to be taken up when the MCT2 is present. So that was our second effort, and that told us that at least this hypothesis of the RPE being able to benefit from more lactate was true. The last thing was to induce the cones to use more lactate. And I don't have time to show you these data, but they were published by Sawyer-Shu, a postdoc in the lab. And we found that if we added a gene called TXNP, then the cones would get off their glucose addiction and start to use lactate as a fuel. And that, again, gave the cones a longer survival time. It gave them greater optomotor. It gave them much better mitochondria as well. So we're not sure the mechanism, but the gene TXNP is interesting. It has multiple protein partners. And one of the things it does is it removes the glucose transporter from the surface of the cones, so they can't take up glucose, yet they live longer. And we found that that's because they need more lactate. We were able to show that with genetic downregulation of the lactate enzyme LDHB. So we know they're using more lactate, and we're not sure the mechanism of how does TXNP induce them to do that. But again, it's a good result. More cones, more better acuity. Okay, so just to summarize what I just told you, by giving the cones and the RPE the ability to make their own in the process of gluconeogenesis, we can get the cells to live longer, function longer. More lactate in the RPE through MCT2. By the way, I didn't tell you, but we only express the MCT2 with a promoter that's active in the RPE. So we know it's the RPE that's using the MCT2. We get more lactate in those cells and longer survival in the cones. And if we use a TXNP delivery, we can get the cones to use lactate. So this is what I'm going to cover on metabolism. But there's also oxidative stress, as I mentioned. In pretty much every disease, there's more oxidative stress. Basically, the amount of ROS, reactive oxygen species that are being produced, which happen all the time in normal cells and increasingly as we age, the cellular mechanisms to reduce the toxicity of those free radicals also goes down with age. So the balance between production of oxidative stress and the anti-oxidative stress mechanisms get out of whack. So as we age, we have more oxidative stress. And in many diseases, including AMD and retinitis pigmentosa, we also have signs of oxidative stress. So we wanted to see if those are part of the cause that causes the cones to die. So we have a couple strategies to fight it. So first, what we thought is, why don't we put in a transcription factor called NRF2, which is the body's normal way to upregulate these antioxidant genes. So it's a transcription factor. It binds to a certain sequence. It turns up a lot of genes, including many of the pathways that fight oxidative stress. It also indirectly inhibits the production of some inflammatory molecules like IL-6 and IL-1 beta. So it's kind of a twofer. It's a benefit in terms of oxidative damage, and it gives a benefit in terms of inflammation. Similarly, PGC1 alpha is a transcription factor that has some of the same antioxidant target genes. Also, it benefits mitochondrial biosynthesis. So we tested them together and separately, and it turned out that NRF2 was very beneficial. And for some reason, we don't know, PGC1 alpha was detrimental to the cones. So we kept following NRF2 then, and here's just some of the data. So this is a retina from one of the RD mutants, and every little green spot is a cone. And what you can see here is in the periphery, we have more cones than in the center. This is one of the disease models with a controlled GFP infection. And we always see a central to peripheral gradient in cone death. So you can see a lot of the cones have already died at this stage in the control virus infected eye. And now we have a lot more cones if we add in an antioxidation strategy. I should mention we also used the enzymes SOD2 and catalase, and they also benefit the cones. But it turns out the NRF2 gives the best benefit. And again, here's an optodrome assay. On the Y, we have the acuity, and on the X, the age. And what you can see in the orange is that NRF2 preserves the acuity much better than a GFP-only control or an untreated animal. So again, a benefit in terms of cone survival and function. We also wondered if it might benefit the RPE. So the retinal pigment epithelium also degenerates in retinitis pigmentosa, and that's shown here with what we call flat mount. So we remove the tissue, the whole sheet of RPE. We stain with phalloidin, which gives us this nice sort of hexagonal regular array in a wild type. And in a mutant, this is the RD1 mutant, it's one of the more rapid ones. We get this terrible change in morphology, and we start to lose the RPE cells. But if we put in the NRF2 transcription factor under control of a BEST1 promoter, which only is active in the RPE, we can preserve it. Oops, So that's what's shown on the bottom right. This is the NRF2 infected, and you can see we've beautifully preserved the RPE, and on the left bottom is the CMV. It's a generic promoter also driving NRF2, and we can also get preservation with that one. So the NRF2 gene therapy preserves the cones, the cone vision, and the RPE itself in 3 different models of retinitis pigmentosa, making us hope that this will be a gene-agnostic therapy. And then we thought, well, if it works so well on those cell types, perhaps it will also work in age-related macular degeneration, particularly the dry form that doesn't have a good therapy. I think most of you probably know what this disease is because it's fairly prevalent, but here's just a little summary. So about 2 million people today in our country are affected by this disease, and it will be more as our population ages because it's an age-related disease. It costs quite a bit in terms of lost value, in terms of people's activities and medical care. And there really isn't a good treatment, which is the dry form. So there's 2 forms. Dry is what I'm going to tell you about. And there's a wet form where there's bleeding in the eye, and that can be addressed at least partially by injections of anti-VEGF, an antibody that stops some of the bleeding. But for dry AMD, which is 90%, we have very little. Now, the way the disease works is still a bit unclear. There are some genetic and environmental dietary factors. For example, smoking is a risk factor. Obesity is a risk factor. For various types of diets are. So we don't know exactly how the disease works because we don't have a great animal model. And we don't have a deterministic gene. That is, you have a mutant in a certain gene and you're for sure going to get it. It just changes the probability you'll get the disease. So we have clues, but we're not in full understanding of the disease etiology. But here's just one idea that oxidative damage is involved. So again, I told you reactive oxygen species are made all the time. And as we age, they outrun the antioxidant defense. And we think they're increasing risk because of several observations. Again, age, genetic predisposition, smoking, and obesity, all of which can affect oxidative stress. And in terms of causality and not just correlation, there is an antioxidant dietary treatment called the AREDS treatment, which can slow the progression from what's called the intermediate to the advanced when there's really a lot of degeneration. And I should say the degeneration is really problematic because it affects our central high acuity vision. I told you in the very beginning about the macula being the area of high acuity vision. For reasons that are unknown, the macula seems most susceptible to age-related disease, and so we're losing high acuity vision as this disease progresses. So this antioxidant treatment works a little bit, but there's a lot of room for a better therapy. Okay, so we thought maybe our AAV NRF2 vector targeted to expression in the RPE might help. There is one animal model, it's very acute and very dramatic, which is to inject sodium iodate, which causes the immediate death of the RPE, which many people think is the site of origin of the AMD. Still not clear, and if I have time, I'll tell you about a model to probe that etiology. But at any rate, what we do is we put in our AAV, BES1 NRF2, again, expressing this anti-oxidative damage transcription factor, anti-inflammation. We put it into the eye, and we use now mice or rats. These are wild type animals. We then inject this sodium iodate or saline as a control when the animals are adults. And our question is, can this AAV, BES1 NRF2, protect the eye from this sodium iodate damage? For some reason, the sodium iodate is really very awful. It kills this RPE very quickly, but it's systemic, but it really hits the eye hard. And then we assay what's happening by ERG, OCT, RPE counts, morphology, all sorts of assays that I'll show you. And what I'm showing you here on this bottom set of panels is this phalloidin stain, again, of an RPE sheet. And on the top left is a NRF2 treated saline injected control. So no damage. We're just putting in NRF2 and it doesn't cause damage. And then on the bottom left is a NRF2 treated sodium iodate animal. And you can still see beautiful hexagons. It's a little bit wonky, but it's still pretty close to normal. On the bottom right is a sodium iodate treated control virus injected animal. And you can see we've lost the RPE. So it's really, again, susceptible. This death happens in a few days. We can quantify. So the NRF2 completely protects the RPE when we have this type of damage. It also protects the cones. So this is, again, a panel of cone counts. And if you look on the right, when we put in the Nerf2 with the sodium iodate, we preserve the cones. If we put in a control virus with sodium iodate, we lose the cones. Saline, of course, has no effect. So we're protecting the cones as well as the RPE with the Nerf2. Now, these are H&E stained paraffin sections. And again, top left is a saline Nerf2. Bottom left is a sodium iodate Nerf2. You can see beautiful normal structures preserved. And on the bottom right is a control virus with sodium iodate. And again, we've lost the structure. We've lost a lot of the thickness of the photoreceptor layer called the ONL. So again, Nerf2 protects the entire photoreceptor layer, which is mostly rods. And if we count cones, it's protecting the cones as well. We also examined this model for leakage from blood vessels, which happens again in wet AMD, as well as diabetic retinopathy, as well as other diseases. And what we can do is inject in live animals a small molecule, fluorescein. Systemically, then, it's throughout all the blood vessels. And if the blood vessels are leaking as a result of damage from sodium iodate, we'll see this diffusion of the fluorescein, which is what you see on the top left. But if we put in the Nerf2, you see we don't have leakage. So the vessels are protected by Nerf2. That has to be non-autonomous because the gene is not expressed in the vessel. So we're interested in how is this protection mediated. If we use an imaging modality called OCT, on the bottom right, you can see this nicely laminated image, which is the normal image, which means that the Nerf2 protected the overall structure. And on the bottom left, you can see a much perturbed structure, particularly in the outer portion where we've lost the photoreceptors and the RPE. So just to summarize, this AAV BEST1 Nerf2 can protect the RPE, the photoreceptors, vessel leakage and death that's been induced by sodium iodate. This is just a little summary of the data I showed you. So we're pretty excited about Nerf2 as a potential therapy for dry AMD. And just to summarize overall, the Nerf2 therapy, we showed that there's less oxidative damage, there's less inflammation, there's metabolic regulation. This is something we gleaned from doing RNA-seq. We're not sure exactly what that means. Lots of different genes change. Those could be secondary or tertiary changes. We don't know. If we express it only in the RPE, which might be the source of damage in AMD, I just showed you we can protect not only the RPE, but the other adjacent cell types. We can also express it in cones and the RPE, which is what we did for the retinitis pigmentosa models. And again, we protected the RPE and the photoreceptors. And we can protect it from this very drastic and acute damage by sodium iodate. So hopefully going forward to some type of large animal model will help. Okay, just in the last few minutes, I want to quickly tell you of a new study. This is all unpublished from Kane Sasaki, who just defended her thesis 2 days ago. So this is hot off the presses. And what we were trying to do is develop a model for dry AMD. This sodium iodate model is very acute, whereas AMD is a very slow disease. And again, it may initiate with oxidative damage, but it at least has oxidative damage. We're not sure where it initiates. So Kane wanted to get a model that allowed us to ask, in which cell type does oxidative damage originate? And can that type of damage spread from the cell type where it originates to other cell types? And I'm not going to have time to show you all her data, but I'll give you the idea. So we in collaboration with Thomas Michel's lab here at Harvard Med School, Thomas's lab had made a transgenic mouse that had a fusion protein of a GFP sensor for peroxide called hyper, fused to something called D-amino acid oxidase. Now the action of DAAO is shown on the top panel there, and what it does is it takes D-amino acids and it creates an alpha keto acid with its reaction mechanism, and that generates peroxide, which is a damaging oxidative stress or ROS molecule. Now, we don't normally have D amino acids. We use L amino acids to make proteins. So a D amino acid is an unusual amino acid. We can feed it to the animals in their water. And if the animal has this construct, this hyperfusion with DAAO, we can then monitor where is the peroxide by using the GFP sensor. And we can create the peroxide by the DAAO only in animals that express this transgene. And the expression is controlled by a Cre-Lox. Some of you probably know if you express the Cre recombinase, that will recombine across what's called the LOX-P sites and turn a gene on. So what we can do then is control which cell type turns on the oxidative damage by using a transgenic mouse that has the Cre only in the RPE, only in the rods, only in the blood vessels, etc. We can choose where to originate the oxidative damage using these mouse lines. We can choose when to start the damage by feeding the animal D-alanine or any D-amino acid in the drinking water. And we can control the speed by the dose. So once we start the D-amino acid, we can change the dose to make it fast or slow degeneration. So our first question was, can we initiate damage in the RPE and then see what happens to the RPE and then see what happens to the adjacent cells, the photoreceptors and the blood vessels? I'm going to skip right to the end, a lot of beautiful data from Kane, and just summarize for you because I'm going to run out, but we use all kinds of assays for this, and we can recapitulate a lot of the AMD hallmarks. So here's what we found is if we turn the gene on in the RPE, we damage the RPE. So here's what we found on the left is an L amino acid control. So the animal shouldn't be generating DAAO peroxide because we're giving it L amino acid. But with D amino acid, we start to lose RPE cells, and so they swell and fill in the empty space. We get RPE structural changes. We've used EM, and the structural changes we see are the same that you see in AMD. We lose the photoreceptors. We can see that by counting the thickness of the ONL or using OCT to do that. We lose cones. Again, we can count the cones on a flat mount. We lose optomotor response. Again, using your wonderful Optodrum, we're losing the acuity. We see blood vessel leakage. We have a couple ways to do that. And we lose ERG responses. So when we initiate damage in the RPE, we're affecting all the adjacent cell types. Now, if we initiate the damage in the rods, we got a really interesting result. At the lower dose, which caused all the damage with the RPE model, we don't get rod damage, but we do get RPE damage. Oh, sorry, we don't get acuity loss, we don't get ERG loss, we don't get vessel leakage, but we do get RPE degeneration. So remember, we're just expressing the DAAO or the oxidative damage in the rods. They're fine at the low dose, but the RPE gets sick. If we raise the dose, we can then get rod degeneration. So this is kind of interesting and unexpected, and we're thinking about what is this mechanism for the RPE loss. And we can also turn the gene on in the blood vessels, and we don't get any phenotype, which is surprising. One model for AMD is that the choroidal vessels have problems, and they're creating something called ghost vessels or dropout of the blood vessels in the choroid, and that's the origin of AMD. But at least here, we get no evidence for that. It doesn't mean that model is wrong. We couldn't keep these animals for a year or so because this gene, when it turns on the blood vessels, leads to death of the animals because it's in all the blood vessels. Okay, so what are we doing in the future? We'd like to test some of these therapies in large animal models. We're testing, and we've already tested now, can the rescues be mediated by later infection when there's a disease process ongoing? I told you we normally are going in before the disease process, and now we're going in later after the disease process has been launched. And we're looking at combinations. Again, as I mentioned, we think there's more than one problem, and would like to develop this as a therapy for both AMD and other diseases, not even just ocular diseases, but other diseases where there's oxidative damage and inflammation. And I'm really grateful to have a number of funding sources that have supported this work. So I'll be happy to take questions now.
Thank you, Connie. Yes, I'm just checking the questions that have been coming in. Give me a second.
Sure.
To get all sorted here. Maybe I'll start with a question that came in when somebody registered for the seminar. So I guess it's more of a general question relating to your work. They ask if in your studies you show that the cones in the RD1 retina at P40 or even later, however, almost all other studies show total death of photoreceptors, rods and cones by P20 or so, with no functional light responses in the scotopic and photopic range. And why do you see longer surviving cones compared to other studies?
Yeah, it's a great question because we've actually shown both things. So it depends on the assay. So if we do ERGs as an assay for rods and cones, you're absolutely right. We lose all function by P21. No cone response, no rod response. If we, on the other hand, use these flat mounts and we count the cones on flat mounts using, for example, an antibody for cone arrestin, we then see cones, and they're mostly in the periphery, but there's still some in the center at P21, and then we gradually lose them. Interestingly, we don't lose the dorsalmost peripheral cones even at 1 year of age. And we had a study that Ryoji Yamamoto, a postdoc in my lab, launched to ask, well, why are those dorsal cones still around at 1 year of age? And he discovered it was due to retinoic acid synthesis by Mueller glia. And now he's trying to understand how that leads to the cone survival and the dorsal periphery. Sawyer Hsu then looked at, well, if the cones are still there at P21, why do we have a flat ERG on the photopic vision? And he was able to track this down. He found that if he crossed the RD1 mutants with a hypomorphic allele of RPE65, so that's a gene that makes the 11 cis retinal, if we have less 11 cis retinal from that hypomorphic allele, now we get back the cone ERG in the RD1 background. In addition, he looked at a mutant for CRA-LBP, which is again part of the retinal handling pathway. Again, we get back the cone ERG. So our model is that the cones are still there at P21, that's for sure from our counting, but there's no cone ERG because they're being bombarded with too much retinal. And the retinal is in overabundance because the rods died. Again, there's 95% of the photoreceptors, so they're not taking up the retinal as they normally would, and the cones are getting too much retinal, which is toxic. So that's our explanation for those kinds of data.
Okay, then there was a question whether your gene-agnostic approach would also be applicable to mitochondrial mutation, in particular ATP6 mutations.
Yeah, I don't know about that particular mutation. We do know that in aging, we're losing mitochondrial function, and in these diseases, we are as well. When we put in the gene TXNP, we get a beautiful preservation of the mitochondrial structure and function. We were able to do functional assays as well as do EM. I don't know if that particular mutation would benefit from just more mitochondria or healthier mitochondria. Maybe not. So I don't really know the answer to the question.
All right. The next question is, why is the macula more vulnerable to degeneration, in particular in age-related macular degeneration? What is it that makes it vulnerable?
Yeah, so I should say we don't know. But here's some speculation, which is, so perhaps it has something to do with the structure of that area. So that area, especially right around the fovea, the inner retinal blood vessels are pushed aside or they never grow in. And so that creates a little bit of difference in terms of nutrient flow, nutrient or waste removal. So maybe it has something to do with the vascular difference there, because the rest of the retina has inner retinal vessels, which again might have something to do with the metabolism of the inner retina. Maybe that affects the outer retina. Again, there's a lot of light. We usually are moving our heads around and our eyes in our heads so that we're getting most of the input that we're interested in to hit that foveal area. That's our high acuity daylight vision. Maybe we're overtaxing that area by using, those kind of sort of preferential reception. That's possible. We've been looking also at the RPE and how it might interface with that area, and there might be different sort of interactions. David Ortolan at NEI showed that there's a slightly different shape of the RPE there. We're also studying how does that area develop. Unfortunately, the typical mammalian models don't have a fovea or a macula, that is rats, mice, etc. So we've been using the chicken, which does have one, and we're studying how does that foveal area develop in the chicken, and we found some genes that might be relevant to susceptibility. So we don't really know, but we're really interested in that question.
Yeah, it sounds you're not running out of questions and out of work.
No.
So what another question is, what is your AMD model that you use to address the origin of AMD?
So the origin question comes from the latest bit of data that I showed you of starting oxidative damage either in the RPE or the rods or the blood vessels. And as I showed, if we started in the RPE, we do affect both blood vessels and photoreceptors.
So in essence, that's a wild type mouse where you introduce your tricks with genetic means.
Exactly.
Mm-hmm.
Exactly. You know, from a perspective of other mouse models, people, you know, sodium iodate again is one of the most common, and that's again a wild type mouse in which you deliver this stuff either directly in the eye or IP injections. And it's not a great model, even though we have used it, because it's very acute and AMD is very slow. Also, you know, those animals don't have a macula, so we're always faced with the idea that when we use these rodents, we don't have a macula, so we can't really recapitulate that part of the phenotype. But this new model we have where we can turn on the oxidative damage at least suggests that the idea that AMD originates in the RPE is a supported idea. I don't think we've proved it, but just that it's a plausible idea that having damage originate in the RPE does lead to photoreceptor and vision loss, does lead to blood vessel leakage. So it could be that that is where it originates in people. Whereas interestingly, the first physiological symptoms in humans is loss of rod-mediated dark adaptation. This has been shown by Owsley and Curcio, and people are now developing it as the primary assay for AMD. It's a functional rod-mediated symptom, and we're not picking up rod susceptibility in what we've done with a low dose. We can get rods to die at a higher dose of D-alanine when we put the DAAO only in the rods. So that's an interesting wrinkle. I'm not sure what it means with respect to the human disease.
Mm-hmm. Then there was a question, at what age and stage of disease do you think your gene-agnostic therapy would still work? Presumably this aims at asking for human intervention with patients.
Yes, absolutely. We think about that a lot because these animal models, everything we've done is fairly rapid models, first of all. And again, we're treating before a lot of disease symptoms. So considering the human case, we have some possibilities. So retinitis pigmentosa doesn't start uniformly throughout the retina. You get pockets or patches of loss of photoreceptors and RPE damage, and then those patches spread. So what we're thinking is if a person is losing cone vision, so again, they're born without night vision, not without good rod vision. When they start to lose cone vision in a local area, the virus would be delivered near that area to put up a wall so the damage doesn't spread to healthier adjacent cones. So this is a prevention strategy. We're not going to cure the already sick cells, we don't think. And certainly anything that's past the threshold of function or good health, I don't think we're going to resurrect dying cells, but we may boost the function longer in really sick cells, but we're also just hoping we stop the spread. Because again, these are non-autonomous disease problems. The cones don't express the disease gene. They're getting sick because of nearby sort of rod-initiated problems. So that spread is what we're hoping to block by delivering the viruses. Now, the timing of that's going to depend on the person and the disease. So I've been told by the ophthalmologist that even a family with the same mutation that causes retinitis pigmentosa can have really different onset of cone loss. So I was told about a family where the brother was losing cone vision at age 20, and the sister was actually a truck driver, and she was still driving her truck at age 60. So somehow, even with the same disease gene, you can have really different pace of the disease and extent of the disease. It could be due to lifestyle differences. It could be due to what we call genetic modifiers elsewhere in the genome. The sister got lucky and got a gene that helped protect. The brother got unlucky, got one that helped destroy, or something like that. So we don't really know, even in the same disease gene family, what the best time to intervene would be. But because the symptoms of cone loss occur over a long period of time, they're not really fast. We're thinking that there will be time to get the vectors in there to the healthy tissue and slow down at least the loss of cone vision.
But would you expect that to be a one-time intervention or repeated injection or repeated expression?
Yeah, so that's a good question, and I would hope we could do it repeatedly. So one issue with delivering a virus as the agent is that you can get immune responses against a virus. Antibodies that might be specific for the viral capsid, for example, might neutralize the virus on its way in if you're doing a second or third inoculation. So Luxturna is the first FDA-approved gene therapy, and it's for a loss of the RPE65 gene that I mentioned, so an RPE gene. They've been able to do both eyes. So initially they just did one eye. And the worry was, okay, after one eye, you won't be able to inoculate the second because it would elicit these neutralizing antibodies, but it turned out to not be the case. The second eye was able to be infected successfully. So it looks like you can at least get past this idea of neutralizing antibodies for a bit of time. I don't know how many inoculations one would be able to do. I mean, theoretically, you can switch capsid types. So you go in with capsid number 1, and second shot is capsid number 2, and third shot is capsid number 3. That's still hopeful, but the problem is a lot of the neutralizing antibodies that have been seen elsewhere neutralize a common epitope across serotypes. So we might not be able to do that strategy, but people are trying. So if we could get around that common epitope, then perhaps multiple injections would work.
Yeah, talking about serotypes, one question was what the most effective AAV serotype is to target the glycolysis pathways of cones and separately of rods. And follow up, when is the best age to inject and to investigate rescue effects of an AAV for RD10 mice?
Yeah, so RD10 is one of the strains that we use for our studies. And we usually, again, are going in at P0 where we can infect everything. Part of the reason we do that for RD10 and RD1 is they're very rapid. And when you infect with an AAV, it takes about 3 weeks for the gene, the AAV encoded gene, to max out in expression. And the RD10 and the RD1 have rod loss very rapid, and the cone loss starts thereafter. So for those strains, we're trying to get the gene in and expressed in time to blunt the loss of cones. And so that's why we're doing it like that. Now, serotypes, we usually use AAV8. It's good for rods and cones and RPE, which is why we use it. There are other serotypes that can also target. There's a few new ones. Companies have been making capsids that are more specific for different cell types. We haven't used any of those. We just stick with AAV8, which works quite well. AAV9 also does. So those are the ones we use.
Okay, thank you. Here's a very general question. What is the current state of gene therapy in preventing blindness? And maybe I'll link with that with another question that's also more of a general question. Let me find it again. What is currently the biggest challenge to develop advanced gene therapy for clinical use? So current state and biggest challenges.
So I guess they're kind of linked. So again, Luxturna was the first FDA approved. It was for a very specific gene defect, which is a loss or reduction in function of one gene, RPE65, and that has been quite successful. There's been some issues with perhaps some sort of toxicity or side effects, but by and large, it's been really safe, really successful. But very few people have that particular mutation. So again, looking at the sort of landscape of mutations that lead to loss of vision, many of them affect very few people. So the cost of going through the clinical trials is hundreds of millions of dollars just per one gene therapy for one disease gene. So that's pretty prohibitive if you can only treat a small number of people. And it's what drives up the cost also per person. Luxturna is really high. Other gene therapies for other diseases where there's few people, very high. So what we need to do is to find ways. Again, I think these generic gene-agnostic ways are important because hopefully they will scale across more people, which will help with the economics of doing the clinical trials. So the expense of launching clinical trials is one big impediment. The second thing is just figuring out the right gene to deliver. As we've actually run through, I showed you some of the winners. We've gone through over 100 genes. I think we're up to 120 genes that we've tested in animal models using exactly the AAV strategy I showed you. And we've got 7 winners. So it's not that easy to find the right gene or genes to deliver. So that's another issue. And I don't know if they're going to translate to people. We think some of these problems we're addressing, like oxidative damage or perhaps inflammation, are in common across mice and humans, but translatability from animal models to humans is always dicey. Although, again, I think it's probably pretty good for the eye because the disease genes we're looking at are the same as in human, and the disease process is the same as we see in humans. So I think we have a good shot at that piece, but it remains to be seen. And then the current status is there are a number of trials going on right now for some gene agnostic, some not gene agnostic disease genes. We'll see. I don't know. I know there's some early reports of some good phase 1 or 1/2 results. So I think there are some that are going to phase 3. I don't think we have any other FDA approved except for 2 complement inhibitor trials or 2 complement inhibitors now that are approved by the FDA. I would say that there's very little benefit to those 2 approved. In fact, they're not even approved in Europe because the benefit is so low and it doesn't give actual functional vision rescue. It just has, there's some criteria that it's met, but that's why we're working to try to get something better. We think that really you want to have better vision. And so that's what we're working towards. But anyhow, that's kind of the current status. People are working pretty hard to get gene agnostic or AMD, dry AMD drugs or viruses because it's a huge unmet need.
Do you mind going like 5 more minutes over because we're already over time, but there's still quite a few questions left?
Go ahead. I'm happy to.
Yeah. So one question was, in what way is your approach better compared to anti-apoptotic interventions, which did not really work well?
Yeah, to me, the anti-apoptotic is kind of, it's just too late. If you've got to the point where you've turned on some of the apoptotic pathways, I think the cell is functionally already dead. So even if you keep it alive, I don't think it's going to function. So again, we're trying to go in at earlier stages by delivering before cone death to try to slow down whatever the causes are of cone death. And as I mentioned, I don't think we're curing as you would do if you were doing a gene editing, let's say. But again, we're hoping it's scalable across different disease gene families. And I think the anti-apoptotic strategies are just too late.
Mm-hmm. Okay. A question about remodeling. How would remodeling hinder rescue, or is it really that much of an issue? Could Miller-Glea take up RPE functions if you have remodeling going on?
Okay, so a couple questions. So how might remodeling affect the gene therapy? Well, again, the thing that I think is important is the stage of treatment. So if you can get the treatment in before the cones die, again, we're not targeting rod death with at least the retinitis pigmentosa models. We're targeting cones, and the cones we're trying to hit before they've died. Now, there will be some remodeling already. Let's say rod bipolars seem to remodel with rod death. I don't know how that is going to affect. I think we just have to go through the experiments with people and see. And the second question was, sorry, remind me, what's the second part of that question?
Oh, whether Miller-Glea,
Miller-Glea.
The specific question was Miller-Glea could uptake lactate.
They could, I think. They have lactate transporters. They produce lactate also. Possibly. I don't know. Again, we would like to be able to track. We were trying to do actually live imaging with the lactate sensor that I talked about. We couldn't get enough expression to do it live. We haven't expressed it in the Mueller glia, which we could do to see how is lactate going in or out of Mueller glia, and that would be a cool experiment to do.
You mentioned combinative approach where you put in several genes and you have additive effects. Do you think that this approach, even though promising, would be prohibitive given the regulatory challenges?
I've been told that. I don't know in the end. I think we have to go forward because my own logic is, okay, if gene X works and gene Y works, and we show that gene X plus Y is safe, why would you stop that? So that's my thought.
Okay. and
I know people are going in. So one more thing, that I know people are going in with dual AAVs, where you have a gene that's too big to fit into one AAV. Yeah. For example, a gene editing kind of strategy. You have to put it into 2 AAVs, and that's being allowed. So that is the same idea, I think.
Yeah, maybe. So the next question, I don't know what exactly it refers to because I don't know when it arrived, but you might know. How do you confirm that oxidative stress actually happened in these mice? A negative result in the OCT and or ERG might just mean the construct or transgenic strategy is not working. I guess it refers to the last project you talked about where it did not have an effect.
Yeah. So we've done a lot of work to try to figure out whether this DAAO is generating either free radicals or oxidized proteins or lipids or DNA. So there's a number of biochemical assays and sort of immunological IHC type of assays. So we've looked for many of them, and we're having trouble seeing a lot of them. We've done, for example, ELISAs for carbonyl groups that get added onto proteins. We see a high background, even in untreated mice, wild-type mice. Apparently, there's a fair amount of oxidative damage going on all the time in the eye, which people have ascribed to high light environment, high oxygen environment, which can create free radicals. So the best stain we've gotten is this molecule you can buy, something rocks. I forget what it's got, some lingo, but it's a small molecule that detects ROS. And so that's what we're using now. We're still trying with these other molecules. Interestingly, for example, the blood vessel DAAO, which doesn't lead to any phenotype, does show some of these modified proteins and nucleic acids as going up when we turn on the DAO there. So there we can see the oxidative damage, but we don't get a phenotype. And the RPE and photoreceptors, the only thing we've been able to see is the ROS small molecule drug. Or it's not drug, but it's a dye that lights up with ROS.
Yeah, there are 2 questions that you maybe can both answer for the DAAO approach in the rods. One is, how did you manage to get AAV into the rods? And number 2, you mentioned that with this treatment, you see effects in the RPE. But nothing else. Would you expect that if you wait long enough with this damaged RPE, you see downstream effects that mimic the AMD that you're looking at?
Okay, first question is, we actually didn't use an AAV. We used a transgenic mouse. So the transgenic mouse will turn on DAAO when we cross it to a transgenic mouse that expresses CRE in rods. So that's how we turn on the gene in rods. Now, subsequently, we've actually made some viruses, some AAVs that have the DAAO under a rod promoter. And we did that so that other people who don't have the transgenic might want to use the virus. For example, if you're working on primates, it would be great to have an AMD model in primates because some primates have a macula. So we're hoping that the viral form that expresses only in the RPE or only in the rods will be useful for other people to use in other species. So that's the first answer. We didn't, in this particular set of data I showed you, use a virus. We used a transgenic. And the second was, if we left it longer, do we anticipate we would get rod phenotypes? Yes. We basically showed that. I ran through the data too fast, but we upped the dose of the D-alanine in the drinking water, and then we get rod degeneration and photoreceptor degeneration, and RPE degeneration. So yes, ultimately we do get that problem. And if we let the animals go longer at the low dose of D-alanine, I suspect we would also get phenotypes in the photoreceptors, but we haven't carried them out long enough. We already did a 90-day assay, and didn't see a phenotype in the rods unless we upped the dose.
Okay, and I think the last question, does your tick SNP work and the reduction in glucose transporter on cones suggest that too much glucose may be toxic to cones?
Yeah, I guess that's a possibility. So we have taken the opposite approach or idea where, again, we think that there's less glucose coming out of the RPE because it's hanging on to the glucose. And I think that's still a little bit more likely than excess glucose. And there was a study from Douglas Dean's lab where they injected glucose into an RP model, and they actually got benefits to the photoreceptors by adding more glucose. And we got more benefits by having the cells make their own glucose. So again, we think that that's the issue, is not enough glucose as opposed to too much being toxic. But formally, that's still a possibility. We tried to, again, image glucose with one of those glucose sensors, and we wanted to do it in vivo, and we tried. We weren't able to get a bright enough signal because we wanted to see, well, where is the glucose? Is it in the RPE? Is it in the photoreceptors? Is it in the subretinal space? And you probably know that these things change within seconds or milliseconds. So when we take the whole eye out to do the imaging, we're worried by cutting off the blood supply, we're going to get the wrong answer. So we haven't done it ex vivo. We've been trying in vivo. But that would be the best way to answer your question. If there's very little glucose, it's not going to be toxic. We think it's just in short supply.
Okay, I hope I didn't miss too many questions scrolling through the list of questions. I want to thank the audience for your eager participation. And of course, I want to thank you, Connie, for a fantastic talk, fantastic science, and your willingness to answer all these questions.
Sure.
I wish everybody a great Christmas time, and I certainly hope to welcome you back next year for more journal clubs, more science, and wish you good luck in the transition to 2026. Thank you very much, everybody. Have a great evening.